Hey everyone, welcome to the Optispan podcast. On today's episode, I'm going to talk about psilocybin, magic mushrooms, and the emerging evidence that psilocybin and perhaps other psychedelics may have a positive impact on the biology of aging. So let's start with why we would even be thinking about psilocybin as a potential longevity molecule. So I would say for the past several years, there has been a growing interest in the potential connection between psychedelics and longevity. And for most of that time, it really felt to me like this was in the area of effects of hallucinogenics on mental health, anxiety, pain, quality of life. Certainly all of those things are important for healthspan and could impact the biology of aging, could impact longevity. But I hadn't really seen much evidence that psilocybin or other psychedelics from a sort of molecular biology perspective were significantly impacting the longevity network, right? The set of proteins and metabolites that interact to seem to regulate the rate of aging, at least in animal models. More recently, there has been certainly a growing interest in the idea that these molecules might be beneficial in the context of specific age-related diseases, particularly neurodegenerative disorders like Alzheimer's disease, Parkinson's disease, et cetera. And there's some preliminary but certainly interesting data in patients with Alzheimer's disease and Parkinson's disease that psilocybin and perhaps other hallucinogenics, at least in certain contexts, can alleviate symptoms of these disorders and maybe even have beneficial effects on cognitive function, at least over the short term. So all of that being said, I was pretty skeptical of the idea that hallucinogenics were going to be very useful in the realm of longevity or geroscience. And honestly, to me, this kind of felt like one of those things where some people sure would like it to be true and they really wished it would be true, but there wasn't a lot of evidence to really suggest that it is true. Then, I don't know, maybe 7, 8, 9 months ago, maybe more than that, I started hearing about some unpublished data in C. elegans for lifespan with LSD or LSD-like molecules. And I thought, that's kind of interesting. And then last month, a paper came out reporting that psilocybin, the hallucinogenic molecule in magic mushrooms, extends lifespan in mice. So I figured I better take a closer look. This is starting to really seem interesting now. So today we're going to do a bit of a dive into that paper. The paper is titled "Psilocybin Treatment Extends Cellular Lifespan and Improves Survival of Aged Mice." It was published in NPJ Aging in July of 2025. Before we dive into the paper, let's start with what is psilocybin? Psilocybin is the hallucinogenic molecule found in magic mushrooms. It acts as a non-selective agonist or activator of serotonergic receptors. Serotonin, of course, is a neurotransmitter in our bodies, and psilocybin is structurally very, very similar to serotonin. So it's easy to see how psilocybin might have an activity similar to serotonin. So what do these serotonergic receptors do? So these receptors are proteins that are found in certain cells in our body, especially in the brain, that are responsive to the molecule serotonin. Serotonin is also known as 5-hydroxytryptamine or 5-HT. When these receptors bind to serotonin, they create signaling cascades inside of the cells that do a bunch of different things. They regulate mood and anxiety, can influence sleep and circadian rhythms, can affect appetite and digestion, modulate pain perception, shape cognition and memory, and even control basic bodily functions like body temperature, blood pressure, etc. So serotonin is a super important neurotransmitter in our bodies that regulates a whole bunch of different aspects of our physiology. Now, of course, psilocybin has very different effects from serotonin. Psilocybin in the body, when you take it, is converted into another molecule called psilocin. Psilocin is even closer structurally to serotonin, and it's the psilocin molecule that actually can bind to these serotonergic receptors, and it does so with varying affinities. So one of the things to appreciate is that psilocybin is a much longer acting molecule than serotonin. Serotonin binds, does its job, and then gets degraded. Psilocin, on the other hand, will bind and it sticks around. And I think that long-lasting component of psilocin is part of the reason why you get these hallucinogenic effects from psilocin and not from serotonin. So another way to say this is psilocin is a non-selective, long-lasting activator of serotonergic receptors. Okay, so non-selective is probably important here, meaning that it can target multiple different classes of serotonergic receptors. And the hallucinogenic effects are thought to be mediated primarily, maybe specifically through a certain serotonin receptor called 5-HT2A. Okay, so it's a specific class of serotonergic receptors that psilocin binds to, and that leads to the hallucinogenic effects. This, in fact, is exactly the same receptor that other hallucinogenics, such as LSD, mescaline, and DMT, bind to as well. Now, it's important to appreciate that if there are longevity benefits, or if there are benefits from psilocybin for, let's say, Alzheimer's disease or Parkinson's disease, that those benefits may or may not be mediated through this same receptor that leads to the hallucinogenic effects. I don't think we have really much information one way or the other on this. So it's just important to appreciate that nuance. So now let's dive into the paper itself. And again, the title of this paper is Psilocybin Treatment Extends Cellular Lifespan and Improves Survival of Aged Mice. Okay, so the first thing I thought when I saw this paper, and again, this is not meant to be critical in any way, it's just one of the things that people who are in science think, is I looked at the journal that it's published in, and it's published in a relatively low impact journal called NPJ Aging. So this is part of the Nature family of journals, but it's not Nature. This is a lower impact journal. And one of the things that I tend to think about is, if a claim like this was really, really rock solid, let's say that the data here are crystal clear, we know the mechanism, there's really no question that psilocybin slows biological aging. That's a pretty big deal. And if the data were really at the highest level of quality, we can be pretty sure this paper would not be published in NPJ Aging. So we go into this, or at least I go into this, with the assumption that this is going to be sort of a mediocre quality from a data or how compelling the argument is perspective. Okay, so that's, before I even look at any of the data, these are the things that are going through my mind. And almost anybody who's got experience in biomedicine or biomedical research is going to have that same sort of an initial thought when they look at a paper like this. Okay, so let's start with the abstract. Psilocybin, the naturally occurring psychedelic compound produced by hallucinogenic mushrooms, has received attention due to considerable clinical evidence for its therapeutic potential to treat various psychiatric and neurodegenerative indications. That sentence is fine. I think, whether the general scientific or medical community would agree that the evidence is considerable for neurodegenerative indications is debatable, but certainly there's some evidence to support that. Okay, so next sentence. However, the underlying molecular mechanisms remain enigmatic, and few studies have explored its systemic impacts. True. We provide the first experimental evidence that psilocin, the active metabolite of psilocybin, treatment extends cellular lifespan, and psilocybin treatment promotes increased longevity in aged mice, suggesting that psilocybin may be a potent geroprotective agent. Okay, so let's break that last sentence down, because this is where the real meat of the abstract in the paper is. So they claim to provide the first experimental evidence that psilocin extends cellular lifespan. So we'll take a look at that. What does that mean? And they claim that psilocybin treatment increases lifespan in aged mice. So we'll take a look at that. If both of those things seem true, then I think it's legitimate to say that psilocybin may be a potent geroprotective agent. Okay, so I'm not going to take you through all of the introduction. They talk about a few things that I think are interesting. They provide what they claim is considerable evidence, talking about greater than 150 clinical studies of psilocybin for a bunch of different endpoints, including psychiatric, that would be anxiety, depression, addiction, neurodegenerative, like Alzheimer's disease, pain, and others. That's all fine. And then they talk about this thing that I hadn't really thought about before. Maybe I'd heard of it, but I never really thought much about it, which is a hypothesis that had been proposed several years ago called the Psilocybin telomere hypothesis. And this hypothesis basically proposes that psilocybin might increase telomere length. And of course, telomere length or telomere shortening is one of the hallmarks of aging. So where does this come from? There's no data to support this that I'm aware of, none in the peer-reviewed literature that I've been able to find, but it was proposed in 2020 in an article in the journal Medical Hypotheses. And the title of that article, if you're interested, is The Psilocybin Telomere Hypothesis, an empirically falsifiable prediction concerning the beneficial neuropsychopharmacological effects of psilocybin on genetic aging. If you read this paper, I mean, it's kind of interesting, but basically it boils down to this. Chronic stress and mental health problems are well characterized to be associated with shorter telomeres in humans. Many, many epidemiological studies have reported this. Psychedelics can improve those things, at least in some people. They can reduce chronic stress and help with at least certain types of mental health disorders. Therefore, psilocybin will promote longer telomeres. So that's the hypothesis. Perfectly reasonable hypothesis, but highly speculative. Okay. So they start the paper by talking about this psilocybin telomere hypothesis, and they suggested that if this hypothesis is correct, then what you might find is that treating cells with psilocybin will lead to longer telomeres. And so they looked in a cellular senescence model where you take cells, fibroblasts in this case, you grow them in a dish, and you look at how many times can those fibroblasts undergo what's called a population doubling. Okay, this is a pretty easy concept. So you have a dish full of cells. At some point, the cells will fill up the dish. They're dividing, right? They're growing, they're doubling. And once they've divided enough times, they'll completely fill up the dish, and then they stop dividing. So cells are sensitive to something called contact inhibition. When they get too crowded, they stop dividing. Okay, so they'll stop dividing, and then what you do is you basically dilute those cells, and you put a subset of those cells into another dish, and you let them fill up that dish. And so if you do that, eventually the cells will stop dividing before they are able to fill up the dish. That's what's called replicative senescence. Leonard Hayflick was the first to characterize this. That's the famous Hayflick limit, when cells can no longer divide to fill up a dish in the laboratory. Turns out they'll do this after the population has doubled about 50 times. And so the way you do this experiment is you grow cells in a dish in the laboratory, and you count how many population doublings does it take before they reach their Hayflick limit or their replicative senescence. Okay, their prediction is, based on the psilocybin telomere hypothesis, that what you should see is a delay in cellular senescence, because telomere shortening is what causes fibroblast senescence in culture. Okay, that's all internally consistent. The problem I have with this is the psilocybin telomere hypothesis is based on the connection between chronic stress and mental health conditions and telomeres. As far as I know, cells in a dish aren't experiencing the kind of chronic stress that a person is experiencing, and presumably don't have mental health disorders. So this isn't really consistent with or a test of the psilocybin telomere hypothesis. Nonetheless, they report a delay in replicative senescence of their fibroblast cultures that are treated with psilocin, the active molecule that psilocybin gets converted into that has hallucinogenic effects. Okay, it's a little weird. I'm not 100% sure I'm confident in these results. One of the reasons is that their cumulative population doublings of the controls flattened out at about 30. Again, typically, this cell line, you would expect to have about 50 population doublings in the laboratory. The way they did this experiment, they started at 10. So even still, the control cells are senescing about 10 population doublings before you would expect them to. And it turns out the psilocin-treated cells that senesce later are senescing right where you would expect the controls to senesce. So, it's a little bit hard for me to have a ton of confidence in this result. But nonetheless, that's what they report. They look at a variety of markers here and see that one of the markers of senescence, something called senescence-associated beta-galactosidase, is reduced in the psilocin-treated cells. The same thing with another senescence marker called P16. So that all is consistent. They sort of immediately jump to this weird hand-wavy hypothesis with SIRT1. As many of you will know, I'm pretty skeptical of the lot of the literature around sirtuins. To me, this kind of feels like, people just testing things that they've heard of in the literature. So I have a hard time putting much faith in SIRT1 being important here. If they really wanted to test it, they could take SIRT1 knockout lines or knock down SIRT1 and shows that the effect goes away. It's sort of irrelevant, but I really don't think there's anything to the whole SIRT1 connection. NRF2 they also looked at and show that this protein NRF2 is increased by psilocin treatment in cells. To me, this is a little more interesting. So NRF2 has been studied a fair amount in the context of geroscience. Molecules that activate NRF2 have been shown to have interesting effects in mice. The NRF2 ortholog is called SKIN1 in worms, a very well-characterized transcription factor that can increase lifespan in C. elegans. So to me, this is a more plausible mechanism. If the effects on replicative senescence are real, this is a more plausible mechanism for how psilocin might be impacting replicative senescence. So in any case, I thought the thing that was really missing here that would have been much more compelling is the obvious experiment, which is to ask whether or not psilocin treatment increases telomerase. Telomerase, of course, is the enzyme that extends telomeres. If, in fact, psilocin-treated fibroblasts have longer telomeres, and that's why they are senescing at a later population doubling, it should be possible and very easy to show that telomerase is increased and telomerase activity is increased. That study was not in the paper, and so I think that was something that probably should have been done that would have strengthened their argument and made me more convinced that this effect on replicative senescence is real. Okay, so all of that is to say that, maybe psilocin can delay replicative senescence of fibroblasts in vitro in a dish. But the real experiment, of course, is to see whether or not psilocybin or psilocin can slow aging, increase lifespan, improve healthspan in animals, which they did. So they got C57 Black 6 female mice from Jackson Labs that were already pre-aged, exactly what they should have done. I guess some people, might argue whether they should have used C57 Black 6. I'm not necessarily somebody who feels like that's the wrong thing to do, so I'm perfectly happy with this design. They started the treatment at 19 months of age, and the psilocybin was delivered by oral gavage, meaning they just kind of squirted it down the throat of the mice. It was given once weekly. They started with a week at a low dose of 5 mg per kg, and then they went up to a higher dose of 15 mg per kg over a 10-month period. One of the things they claim in the paper is that they saw qualitative improvements in fur quality and hair graying. The pictures look pretty interesting. Again, it's qualitative. It's a little bit hard to know how much faith to put in this, but they do show before and after pictures of what are apparently the same mice. It kind of looks like psilocybin is causing the mice to either grow new fur or have their gray fur turn into black fur. That's pretty interesting if that's real. So kind of intriguing. And then the lifespan data. And if you look at the lifespan data, there are some limitations to this data set, which again may be part of the reason why this was published in NPJ Aging rather than Nature. One of the limitations is that the experiment wasn't actually completed. So they stopped the experiment, or at least they went to publication when the control mice reached 50% survival, or their median lifespan, which was just short of 28 months. at that point, only about 20% of the psilocybin treated mice had died. So 80% were still alive. So, it's an incomplete experiment, but it looks pretty good. And you can certainly do a statistical analysis to determine that median lifespan is increased in the psilocybin treated animals compared to the control animals with a reasonably good p-value of 0.014, well below the 0.05 cutoff. So that looks pretty good. And my eyeball test here is, maybe the controls didn't quite make it to the 900 days. Pretty darn close though. And this looks real to me. But the thing I always like to do then is go back to other experiments where a different intervention that's been reproducible has increased lifespan and ask, how does the data here look in an absolute sense compared to the data from these earlier gold standard studies? And one of the studies I like to go back to is a paper that we published from my lab in 2016 in eLife, where in both male and female mice, we treated them with rapamycin at different doses for 3 months, between the ages of 20 and 23 months of age. And in fact, in that study, we used C57 Black 6 mice from Jackson Labs. So it's a perfect apples to apples comparison here. And we can just overlay the data from this report with the data from that study. And so in that study, in the female mice, we saw a modest but statistically significant effect of rapamycin. The rapamycin effect was bigger at this dose in the males, and it was even much greater in males at a higher dose. But nonetheless, in the females, we saw a pretty good effect of rapamycin. We put those curves on top of the curves from this study, and what you can see is the data in this study look pretty good. That seems comparable, at least up until the point that they stopped the experiment, with our data with rapamycin. So to my eye, given the limitations to the incomplete experiment, it looks like a real effect. So I'm kind of starting to believe that psilocybin increases lifespan in mice, which is really interesting. So I sort of feel like there's a pretty good chance this will reproduce in subsequent studies. Hopefully those studies will get done. I think, unfortunately, they won't likely be done by the interventions testing program unless somebody can formulate and demonstrate bioavailability of psilocybin in the chow. This is more a feature of the way the interventions testing program does these experiments, that the drug needs to be formulated in the chow in order to be put into the pipeline in the vast majority of cases. So hopefully somebody will reproduce this study, and it would be great if it could be done by the interventions testing program. But my gut feeling is it's probably a real effect. And so all in all, I mean, I understand now why this paper was in NPJ Aging and not in Nature. There are some weaknesses that maybe limit the interpretation a bit. I already talked about the fact that I feel like they kind of tried to jump on, stuff they'd heard about, looking at SIRT1, looking at telomeres. They didn't actually do the key experiments to test whether or not those factors are involved in at least the effects in cell culture, which would have been pretty easy to do. So that reduces my confidence a little bit. But again, the mouse lifespan data looks pretty good. And so that, balances out that skepticism I have. So let's take a little foray now into what might be the mechanistic explanation here. This is going to get speculative because we don't have the data. They didn't actually do any experiments to really get to mechanism. So let's start with SIRT1, the one I think is least likely. The only real rationale for SIRT1 being involved are based on studies from Shin Amai's lab at Washington University in St. Louis, where they created a mouse called the Brasto mouse. This is a brain-specific overexpression of SIRT1. And they reported that that mouse is long-lived. That paper was published back in 2013, so 12 years ago now. And as far as I know, nobody has ever replicated that. I'm not saying it's wrong. I'm not saying people have tried and failed to replicate it, except there was at least one other Brasto line that did not show lifespan extension. So even within the same lab, there was a failure to replicate the lifespan extension from this type of brain-specific overexpression of SIRT1. So I'm sort of like, maybe not that the Brasto mice are long-lived. Outside of that, there's not really any good reason to believe that SIRT1 might be involved. So their model, the author's model, is that psilocybin is going to increase SIRT1 in the brain, and based on the Brasto mice, that could promote increased lifespan. So that might be how psilocybin is acting. Again, I already talked about the fact that Brasto maybe not long-lived, but it turns out that the evidence that Brasto SIRT1 is acting through serotonergic signaling, not the other way around. So the directionality of serotonergic signaling here is opposite of what you would expect based on this model. In other words, the only evidence that's available would suggest that SIRT1 overexpression is leading to more serotonergic signaling, which is in principle the same thing that psilocybin is doing, not that psilocybin would lead to increased SIRT1. And in fact, we know that increased SIRT1 is not necessary for psilocybin to lead to increased serotonergic signaling because psilocin binds directly to the serotonergic receptor. So the model just doesn't make a lot of sense given the biology that we know now. Again, the obvious experiment here would be to see if psilocybin can increase lifespan in the SIRT1 knockout mice. That's a hard, long experiment. I don't fault the authors for doing that. So at this point, not much reason to believe, in my view, that SIRT1 is involved here. I think the telomere model is more interesting. So if psilocybin is turning on telomerase, then that could be sufficient to account for the lifespan extension that they observed. Their cell culture data, if real, is certainly consistent with the idea that psilocybin activates telomerase. Again, it's unfortunate that they didn't measure that. That's an easy experiment. Somebody should do that. Hopefully somebody will do that. And if that turns out to be the case, then I think that suggests a pretty compelling hypothesis, which is that psilocybin turns on telomerase, and that's leading to the increased lifespan. There's pretty good data from Maria Blasco's lab that, at least in certain contexts, activating telomerase in mice is sufficient to increase lifespan. So that's a plausible hypothesis that's very testable. Another model that I thought was intriguing is a connection to serotonergic signaling. And we know that serotonergic signaling in C. elegans can increase lifespan. So multiple studies have connected mutations in the serotonin signaling pathway or in the receptors themselves to lifespan extension in worms. Now, again, doesn't line up completely because much of the data in worms comes from what are called loss of function mutations in serotonin receptors or components of the serotonin signaling pathway. There's also a drug called meancerin that also targets the same receptor, 5-HT2A receptor, and increases lifespan in worms. So a little bit of lack of clarity around directionality, but the fact that the same pathway, and in fact the very same receptor, has been implicated in longevity in another animal model, in C. elegans, suggests to me that there's probably a there. And so I think some work around understanding how this is working in C. elegans and how that may translate to mice could be beneficial. In fact, there's pretty good evidence from worms that serotonin signaling can interact with the worm skin-1. And you recall, I mentioned skin-1 is the NRF2 ortholog in worms. And in this study, they reported that NRF2 is activated by psilocin in cells. So again, a connection that could be explored in mice. One obvious question would be, is NRF2 activated in a variety of tissues and organs in the mice treated with psilocybin? So again, the model here would be psilocybin gets converted into psilocin in mice. Psilocin binds to the serotonergic receptor, and that leads to increased NRF2 activity, and that leads to increased lifespan. So again, very testable model, a variety of experiments that could be done to flesh that out. And then the final connection that I also thought was interesting is a connection to BDNF. BDNF stands for brain-derived neurotrophic factor. BDNF enhances synaptogenesis, brain plasticity, It's known to be induced by psilocybin in both cell-based and in animal studies. There's a lot of interest in the field around BDNF and cognitive aging and cognitive function during aging and dementia. And at least in mice, hypothalamic BDNF gene therapy, so overexpression of BDNF in the hypothalamus, a region in the brain, is sufficient to improve metabolic health. I don't think there's data on lifespan yet, but certainly plausible that that treatment could have a positive effect on longevity as well. So that suggests yet another way that psilocybin could be working here by increasing hypothalamic BDNF and thereby increasing longevity. These are obviously not mutually exclusive. It could be more than one of these things. It could be some combination where these pathways are interacting. So I think there are a variety of potential approaches that can be fleshed out in the future to really dive deeper into the mechanism. So all in all, I'm pretty optimistic here that psilocybin can have positive effects on longevity, at least in mice. Certainly would not suggest that people go out and start taking psilocybin because they believe that it is going to increase their lifespan. I think maybe we'll do a longer episode around psilocybin, hallucinogenics, and their sort of broader role in health, potentially longevity. But at this point, I think this is interesting. And I certainly believe that there is a good rationale for future studies to figure out how psilocybin is having these effects. Does it require the hallucinogenic aspects of psilocybin, or might you be able to get some of these other benefits, apparent benefits for longevity, metabolic health, brain health, without the hallucinogenic effects? I think all of those are super interesting questions, and I'm looking forward to hopefully getting some of the answers. Okay, so that is my take on the recent psilocybin longevity paper. And as always, I hope you found this interesting, informative, maybe entertaining. I don't know. And if you've got any questions or comments, I would encourage you to either leave them here or go to our website, www.optispan.life/podcast. You can leave any questions or comments there. We read all of them. We often attempt to answer them on future podcast AMA episodes. And if you're not yet a subscriber, I'd encourage you to hit that subscribe or follow button below. And I hope to see you next time on the podcast.